Hello, good evening, friends. So let's start the session. So my name is Ankit Kumar Malik, NPTEL PMRF Fellow, or PhD scholar from the Department of Pharmaceutical Engineering and Technology, IIT BHU. And the course is gel-based proteomics and sample preparation for the day. And this is the second week for the same course. So the topics which we will cover today are system biology, sample preparation of proteomics, and one-dimensional electrophoresis. So let's start with the system biology. So after discussing proteomics, now let's talk about systems biology. So what is systems biology? Is that an historic knowledge, a method to understand biology systems, or a tool to solve the practical problems? Okay, so let's start. So what is system biology? So system biology is an examination of a biological entity as an integrated system rather than the study of its individual characteristic reactions and its components, which is termed as system biology. So, a study of all the mechanisms underlying the complex biological processes in the form of integrated system of many interacting components is studied under the system biology. So, the system level understanding of biological networks requires information from different levels. So as you can see from DNA to RNA to proteins forming systems, and then that information can be applied to understand a complex system for different organisms. So the biological information is represented by the networks of interacting elements and dynamic responses to the perturbations. Okay, so these networks provides insights which cannot be analyzed from the isolated components. The systems, then the common elements of the systems biology includes networks, then modeling, computations, and dynamic properties. Okay, so the next is the biological networks. So it covers the protein-protein interaction networks, gene regulatory networks, protein DNA interaction networks, protein lipid interactions, and metabolic networks. Then the various ingredients of systems biology. For example, if you are studying about a cell and the system's behavior, so one need to look at the genome, its transcriptome profile. Okay, so proteome profile, how protein DNA and different type of transcriptional networks are altered, protein signaling networks. Okay, so multi-array complexes, how they are formed, how protein is localized in the intracellular dynamics and metabolic networks. So all of these ingredients of a study about a system. So now systems biology study can be done at different levels. For example, to study the complex physiology of human, one can look at the individual systems such as respiratory systems, nervous systems, or other physiological systems. So the studies can be done at the intercellular or intracellular level, and finally at the molecular level involving genomics, transcriptomics, and proteomics. Okay. So the next is what is the aim of systems biology. So to understand the biology in holistic approach rather than the reductionist approach, so the system biology aims to quantitate the qualitative biological data and provide some level of predictions by applying different type of computational modeling. So why do we need systems biology? What is the need for systems biology? So the study of biology at the system and subsystem level for understanding the biological processes and network is much required. As you can see, to understand even simpler system of a cell, how it is regulated with its extracellular space, the cytoplasm and different other components, examination of the structure and dynamics of cellular and organisms function, so which is very much required for the understanding of the system rather than the characteristics of isolated parts of any cell or organism. Okay, so the systems, there's one more thing, the systems biology approach. So which involves first of all the collection of large experimental data sets and then mathematical models to provide insights of some significant aspects of the data sets. So the simple systems biology approach would involve experiments by adding new data sets which will be used for model constructions and model analysis and the biological insights which is derived from any models can be used to propose new hypotheses. So the properties of a system are probably more than just the sum of all its individual properties or its components. So therefore, it is possible that system may have its own property by applying all the components. So what are the different approaches have been taken to study the systems biology? So the six distinct approaches of systems biology include the model-based and data-based methods in the model. Whereas in the database objectives, it is to find a new phenomena. The model base relies on computational modeling and simulation tools, whereas the database methods relies on the omics data sets we say. Then so in the model based systems, biology approach is difficult to build the detailed kinetic models. So, but in database system, the complex relationships among the various types of omics information and metabolic pathways and networks, so which can be created by studying different systems component in a very challenging systems biology and their biological networks. So, which aims to understand the system structure and functions for better understanding of system property. Okay. So the next thing is systems biology triangle. So what is systems biology triangle? First of all, the systems information is generated at various levels. So as we have discussed, starting from genes, to mRNA, to proteins, to metabolite, as you can see in this diagram, or identifying the regulatory modifiers, metabolic pathways, and functional modules, and different large-scale organization. So this information has to be stored, processed, and further executed to identify the system level information. So even at a simpler systems such as cell can be linked with various properties as you can see in this diagram. So it's genome sequences of different molecules, intracellular signal transcription factors, different types of cell binding activities, the expression profiling of RNA and proteins, and a different type of cellular processes. Okay. So there's one more system biology triangle experiment. So the systems biology triangle, as you can see here, which involves the experimental data sets, which could be derived from different types of omic platforms technologies. So how the computational analysis can be performed, different types of bioinformatics softwares and tools that can be computational modelings by obtaining some theoretical concepts. So thus energetic applications of the experiment theory and technology, which modeling to enhance the understanding of some biological processes as whole system rather than its isolated part is termed as a system biology triangle. Okay, so system in this diagram, the system biology triangle, the wet lab experiments are bioinformatics based data analysis can be used to propose a model. So the model building as an aid to understand the complex system and some hypotheses can be generated, so which could be used further to propose more quantitative models or predictive models. And also it can be used for independent techniques for the model validations. So the next topic which we'll cover is sample preparation for proteomics application. So I'll give you the overview first. So welcome to the proteomics course. Actually, this is the same different topics. So welcome to the proteomics topic. So in this lecture, so we'll talk about the sample preparation for proteomics applications. As you know, a very good sample is essential to perform any good experiment. So, and especially when you want to characterize thousands of protein in a given experiment. So it is very essential that you start with a very good sample. Okay, so an ideal sample preparation steps ensures that you have no contamination and you have a very good protein yield. So and no interfering substances are present in your extract. So often, many times, so each type of biological sample poses its own challenges. For example, if you are working on bacteria culture, working on plants, working on some human samples, so different body fluids such as serum, urine, saliva, spinal fluid, each sample types bring its own unique composition and challenges. So then you are performing sample preparation. You have to be very cautious that what sample you are processing. You cannot follow a generic protocol. So you have to optimize the conditions depending upon your sample. So an ideal sample preparation will ensure that you have all the protein present in the sample without different type of contamination. So contamination is a very important factor here while preparing the samples for proteomics or protein extraction. So different contaminations such as nucleic acid salts and other interfering components. Okay, so now here I'll give you a workflow for protein sample preparation, such as the first step is to disrupt the cell, how to lysis them, how to protect the cell during the lysis step so that there is no proteolysis occurring, how to fractionate the sample. Often you need to simplify the proteome. You need to ensure that the sample is simple enough to show the good protein throughout the proteome coverage. So to obtain a comprehensive proteome coverage, so often it is very important that you should be pre-fractionate your samples and protein extraction followed by solubilization. So these are another very essential components. So now when we are talking about the sample preparation, so this is quite generic for different type of proteomic application, whether one want to use for gel based approaches or gel free approaches. So whether you want to use two-dimensional electrophoresis, dyes, or different types of gel-based applications, or you want to perform some mass spectrometry and then different types of label-free techniques as well. So you have to ensure that you are starting with a very good protein extract. But cell solubilization and some other components have added more when you are performing the gel-based proteomics. Okay. So the next is the sample preparation proteomics, the continuation of the slides. So here you can see proteomics is surrounded by some organism, tissue, fluids, and cells. So the proteome is a very complex. Whether you want to perform a proteomic analysis for the whole organism, it means you want to know all the proteins present in a given organism, or in a tissue, or in a body fluid, or in any very different types of cells. So proteomics can be globally or it can be very targeted or expression based. So you are highly reproducible samples. So which is very important for performing comparative and proteomic analysis. And if you want to know how the difference in your sample as compared to the controls, so you need to ensure that your sample preparation is very reproducible. If you introduce some facts to begin with, then obviously not going to identify the reproducible biological changes. Okay. Then, as you can see here, some global proteomic analysis, expression proteomic analysis, and targeted proteomic analysis are there. So, these are the three different terminologies here for proteomic analysis. So, we'll start from one. So, which is a global proteomic analysis expression. So, this expression proteomic analysis and targeted proteomic analysis. When I'm talking about global proteomic analysis, so it means your aim is to characterize all the protein present in the given sample. So expression proteome analysis, so it means you are mainly interested to look for those changes which are big due to any chemical or a treatment. Those are induced either going up or down, the protein amount is changing. Okay. So the protein expression analysis that is most commonly used for various types of clinical and different studies. And the last one is targeted proteomic analysis. So if you are very focused for a given original or a given sample type, often you would like to know what is happening in that particular proteome. So, for example, mitochondria proteome is there. So, one need to try different type of strategies. So, one think about performing a sample preparation. So, what is your objective? Whether you want to do global profiling or you want to do expression profiling. In either case, you need to accept all the proteins present in that particular target sample. Okay. So now when you are looking at a targeted proteome analysis, you just want to pre-fractionate your sample. So in such a way that only that particular component is isolated, and then all the proteins from that original or originally or cells which is from any data sets is extracted. So different types of strategies need to be used to perform these types of proteomic analysis. Okay. So now all the samples processing involves solubilization, denaturation, reduction, and the treatment of sample proteins. So, but you need to involve additional steps depending upon the type of samples and your type of objectives, so that the protein quality, the protein extract can be improved. And while you are doing this, so you have to be very cautious that when you are performing various steps and the sequential way. So, type of extraction, you may also lose a small fraction of the protein. So while performing this experiments, you can lose. So one has to be very careful when adding various additional steps during sample preparation. So now protein extraction protocols, they need to ensure that most, if not all, the protein in a cell, it's originally and extracted the presence of the interfering components or a compounds. So should be minimized. So if you have optimized in a very good protein extraction, so procedure that should ensure that you have a very wide proteome coverage, and that is ultimately going to determine the success of your proteomic experiment. Okay. So the next thing is ideal protein preparation. So ideal protein preparation involves solubilizing all the proteins present in a given mixture, which prevent protein aggregation, denaturing, and reducing all the proteins which are present. Ideal protein preparation in that mixture removal of nucleic acid and other contaminants, as well as removing salt and some other small interfering components. So again, depending upon the sample type, you may have to think what a different type of interfering components could be present in that sample. So if we are talking about plant roots, you may have to get rid of phenolic components. Similarly, you have to think specific sample types and what could be the major contaminants present in that particular sample. So for example, serum which is rich in a lot of soluble components, you need to get rid of those things. So an ideal protein preparation should involve all of these steps as I mentioned previously. Okay. So next is some guidelines. So guidelines for sample preparation. So here I'm giving you some guidelines for sample preparation. Starting point one should be start with finding a good reference from the literature as the guidelines for sample preparation. So starting point and then try to modify the protocols depending upon the objectives. So that experiment you need to ensure that you remove nucleic acid salts and different particles, prepare the sample as freshly as possible and store it in -20 degree in a very small container. So you should avoid repetitive freezing and thawing of a sample. Okay. So the next is workflow of sample preparation. So here I'll discuss all of these points one by one. So this is a workflow of sample preparation which involve first some cell disruption analysis, cell disruption lysis. So second, protection from proteolysis. Third one is sample fraction. Fourth is protein extraction and solubilization. Fifth is contaminant removal, and sixth is quantification. So I'll cover this one by one. So I'll go in on very basic things and very basic principle of these workflow uh, topics. So obviously this is a workflow can be modified depending upon your sample size, and few steps can be moved in that sequence. So let us follow this workflow, and during this workflow I will give you some examples of different types of samples. So how to extract protein from those, and what type of challenges these samples are going to impose. Okay, so the first one was cell disruption or lysis. So why do we need to do lysis? So to facilitate effective disruption of cells or tissue, or to isolate proteins from intact cells and tissues while avoiding loss or modification of proteins. Okay, and also to obtain all the proteins in the sample, which is not possible. You can lose some amount of protein as well. So this lysis helps to maximize the sample recovery, retain structural integrity of the sample. And the second one is protection from proteolysis. So which means cell lysis releases some proteases, which may result into proteolysis. So during the sample preparation, proteases inhibitors are added to minimize some artifactual proteolysis. So I'll give you the example of those protease inhibitors. So effective protease inhibitors contain a mixture of irreversible or reversible proteases inhibitor that inhibit serine, cysteine, and some metalloproteases. Okay, this was specifically used for some gel-based proteomics. And the next was sample fraction. So a fractionization makes it possible to isolate groups of proteins or fractions from the total proteome, which simplifies the analysis of complex protein mixtures. So this allows for improved resolution when an individual fraction is analyzed. So this provides some less crowded 2D maps. Okay. So what is the importance of the fractionization? So the fractionation increases the proteome coverage. So this separates highly abundant proteins from low abundant proteins of interest and bring them into dynamic detection range. So and also it increases some chances of identifying low abundance proteins of diagnostic or therapeutic interest. Okay. So the last topic is one-dimensional electrophoresis. Okay, so we left this protein extraction. Sorry, the fourth one is left. The contamination removal and quantification I've already discussed in the previous slides. Protein extraction, solubilization was left. So I'll cover this also. So the protein extraction preceded by some subcellular or fractionalization to enrich proteins of interest. So some protein extractions in aqueous buffer, like example are tris HCl acids and followed by desalting it. Then protein precipitation by TCA we called, which is known as trichloroacetic acid, and or acetone we can say. Then TCA and some acetone, some analytical type of acetone. Then the protein extract should be soluble, free from protein, then DNA, RNA, protein interactions, some metabolites. Okay, so the fifth and sixth contamination removal and quantification I've already discussed in some previous slides. So the next topic is one-dimensional electrophoresis. So the one-dimensional electrophoresis, wait, so the one-dimensional electrophoresis, the separation is based on the charge to mass ratio and the molecular weight of the protein. Therefore, on a given gel, if you have a smaller the proteins in the electric field, they will move further down, and the higher molecular weight which remains on the top. So if you apply some standard molecular weight markers, so which give you the known molecular weight of that particular protein. So then for a given unknown protein, you can determine the molecular weight, and it is expected that the higher molecular weight proteins will remain on the top and smaller molecular weight proteins will reach towards the bottom of the gel. So commonly employed one-dimensional technique includes sodium rhodocyl sulfate. In common language, we say SDS page. Okay, or other technique which is used for studying the protein in the native conformation native form, which is known as the native stage. This is polyacrylamide gel electrophoresis. Okay, so this is the electrophoresis for protein characterization. So the electrophoretic technique can be used for the protein characterization to study different properties of, for example, the subunit composition, as you can just both characterization is given here. So the first one is the subunit composition of the proteins. What is the molecular weight of those subunits, and what is the molecular weight of the native protein, and different type of post-translational modification. Again, only the one technique which may not be able to answer all of these questions. So often you have to involve more than one type of electrophoretic technique in that. So for example, while comparing native and subunit composition forms, you need both native electrophoresis and SDS page. So if you want to look for the post-translational modification, then SDS page will be useful. So if you want to compare the proteins based on the isoelectric point as well as the separation of the isoform or some PTM form, so based on the molecular weight, again 2D electrophoresis will be useful. So no one need to know the concept and the laboratory way of using these techniques, so that by applying the combination of the technique can be study different type of properties. So now I'll discuss uh, some in brief manner about SDS page. It is sodium rhodyl sulfate polyacrylamide gel electrophoresis, which is a technique widely used in the biochemistry, forensic genetics, and molecular biology to separate and identify proteins according to their molecular weight. So this method separates proteins based on primarily on the molecular weights, as I've told you in the 1D technique. Okay, so this is a concurrent treatment with a disulfide reducing agent such as beta mercaptoethanol, or we can say DTT, dithiothiotol, which further denatures the proteins by reducing disulfide linkages. So thus overcoming some forms of tertiary protein foldings and breaking up tertiary protein structures. So the proteins samples are having uniform structure and charge. So the separation will depend on their molecular weight only. And some small proteins migrate faster through the gel under the influence of the applied electric field, as I told you now. So thereby in the electric field, protein molecules move towards the anode, which is positive one, and separated only according to their molecular weight, as you can see in this structure. So the first one is which is not in the uniform manner, but was of before the electrophoresis, and it, after SDS it got in some particular uniformly manner. Okay. So the protein samples are having uniform structure and charge, then separation will depend on their molecular weight only. So sodium cortisol sulfate treated proteins have very similar charge to mass ratios and very similar shapes. So during page, the rate of migration of SGS treated proteins is effectively determined by the molecular weight, and the small proteins migrate faster through the gel under the influence of the applied electric field. So whereas large proteins are successively retarded due to the sieving effect of the particular gel. Okay. The next is uh, the page, which I've already conjugated with the SDS and page. These are different principles and some different definition for the page. So the page is stands for polyacrylamide gel electrophoresis. So which is formed by the copolymerization of acrylamide and a cross linking by NN-methylene bisacrylamide. Okay. So to polymerize the gel, a system consisting of ammonium persulfate, which is an interior, and a tetramethylene ethylene diamine, which is called TMED, is added, which acts as a catalyst here. Okay, so this is the structure and the figure for the formation of polyacrylamide gel from the acrylamide and bisacrylamide. So there are some SDS page preparations for the first step which we have to look into is the sample preparation. So here I have given some protocol of my experiment. So in which I have taken some 40 microliter of protein samples, which was added with them some 10 microliter of disruption buffer, then followed by boiling it to the mixture of 3 minutes at 99 degrees Celsius. So how I prepared the disruption buffer? It contains so these kind of things, chemicals. So I cannot give you the entire protocols and how much amount I have taken. Okay, so I've just given the names and the of the chemicals. So the second one is page preparation, polyacrylamide gel preparation. So so here cross-linked polyacrylamide gel are formed from the polymerization of acrylamide monomers in the presence of smaller amounts, which I have told you in the previous structures. So preparation separation gel preparations contains these stock solution chemicals, and the volume of stock solution is required to make 12% of polyacrylamide gel, and these are the some parameters given here which I have taken from my experiment. And this then another thing is stacking gel preparations. So the third step is running the gel using some running buffer at pH 8.4, which contains these chemicals glycine, SGS, and triacethyl, and followed by the staining the gel using some staining buffer, which contains some glacial acetic acid, methanol, and Cosme Brilliant Blue 50R, and followed by the destaining the gel using some destaining buffers, so which contains glacial acetic acid and methanol. Okay, so this is all for today. So in the next session we will uh, talk about some 2D electrophoresis. So in today's we have covered the 1D electrophoresis. So we'll meet you in the next Friday, in the next section. So you guys can leave the session. Thanks for the day.